it really comes back to the point I made about treating the temperature profile as like any other aspect of your assay. If you have variability in the way you do that, then you will get variability in the results. And the way that modern technology can help is that, for example, a controlled rate freezer, of which there are several companies who make those with different technologies, but the fundamental point is that it's electrical. So the rate of freezing is electrically controlled.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Are you wondering what the best cryopreservation strategies and technologies are? You are in for a treat today because I'm having a conversation with Mark Davison, who is an experienced entrepreneur and is now the CEO of Grand Instruments, a leading company in laboratory equipment. Stay tuned. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Mark, to the Smart Biotech Scientist podcast.
Thank you very much, David. Great to be here.
Yeah, it's a pleasure having you on today, Mark. Mark, tell us, how did you get started in biotech and how did you arrive at your current CEO role?
Wow, that's a longer story than some people. Given my lack of hair, you can guess that one. But the original piece is really easy. So probably like many people listening, I had a hugely inspirational teacher at school. I think most people can trace the fire spark back to somebody. In my case, it was a guy called Mr. Carter, who was my high school biology teacher. There should be a Nobel Prize for teaching, I think, that for some of these guys are so influential in future careers. And this guy just opened my eyes to the potential of biology as a discipline. And high school, obviously, it's not molecular biology, but it's that width of plants, animals, how things live, how they die, how they reproduce. And that kind of led me into a degree in biochemistry, which back then was simpler than it is now. I remember vividly, and during my degree, the PCR excitement was coming out, Carrie Mullis, like the original PCR excitement. That makes me an old man. But that kind of excitement was there in the late 80s, 90s, when it was the molecular biology revolution, the Human Genome Project was just starting, that whole area of change of biochemistry from a really long process where you did everything with manual purifications and things took weeks and a whole PhD could be spent just purifying a protein, to speeding the whole thing up and really opening everybody's eyes to the possible imagination almost of what could be done in biology. That's where my kind of original start became. And then I joined pharma company. So I joined what was, when I made the application, it was Beecham Pharmaceuticals. It then became SmithKline Beecham and then GlaxoSmithKline a few years later. And I just wanted to make a difference, right? So the theoretical biology was great, but I wanted to apply it to something interesting. I went for the interview. I actually went for the interview at a place where the address was the Chemotherapeutic Research. So I prepped a whole lot of stuff on cancer and oncology. And I got there and it was an antibiotics research site. So I had to quickly think on my feet and realize that a different chemotherapy. So I ended up spending 7 years researching antibiotics and how do we kill bugs? How do we kill bugs but not kill people? That turns out to be the slightly harder trick. Can kill bugs with many different things, but most of them will not be very nice if you swallow them. So that was great fun. And I'd probably still be there were it not for one of the many twists and turns of corporate life where suddenly your role is laid off with hundreds of other people. And then I kind of journey then through corporate life. So that started my commercial career really around selling into and on behalf of the life science industry. So everything from virus testing for cell lines through to eventually clinical trials and actually even software in the supply chain. But always with that life science kind of sun at the center of my solar system, if you like. So different ways around that. And then most of my career was accidental. If you get an honest opinion from most people, very few people design how they are in their 50s. Usually it just happens to you while you're trying to do something else. And in my case, that's certainly been the case on 2 or 3 occasions. And here I am now. So now I run a mid-sized company making scientific instruments in the UK. And all of those kind of checkerboard experiences give you a good wide perspective in doing this kind of role.
Wow, I love your story, Mark. And like you, I had a teacher in high school who was pivotal for me. In my case, he was my chemistry teacher. And then I started my chemical engineering studies and moved into biotech. His name was Mr. Lipscher. And I think it was because of him that I decided to study and now become a biotech engineer and scientist.
Yeah, and I bet if you talk to your classmates, there's probably 3 or 4 people who also derived their career from the same guy. And I know it was certainly in my case. And 2 or 3 other teachers as well. People have inspirational dance teachers or whatever it might be, but it really does make a huge difference.
Absolutely, it does. Teachers are important persons. Now, Mark, let's talk about our main topic today, cryopreservation. And I would say the biotech revolution wouldn't be what it is today if we hadn't developed all these powerful tools to preserve cells. And now I'd say it's becoming even more important with all the new modalities and cell therapies and you name it. And there's a lot going on. So could you explain our listeners the critical role cryopreservation plays in preserving cell viability?
Sure. And it's really boils down like most things to water. So if you imagine the cell is a very fragile bag of water, you know what happens when you put a tray of ice cubes in the freezer, the water expands over the top of the tray, right? So the volume of the ice is more than the volume of the water. If you're a cell, then that can cause the cell to burst and therefore the cell is inherently dead. So cryopreservation is really around putting cells to sleep in a way that you can wake them up again and they will grow. And it's really as simple as that. And depending on your cell type, that can be relatively straightforward. Some cells are relatively bulletproof. You can be rough with them and they'll survive. Bacteria tend to be pretty robust within reason. Some cells are really fragile. And if you do anything wrong in both freezing and thawing, then you end up with a poorly viable cell population. And then when you move up the chain into things like organoids or embryos or that kind of multicellular apparatus, then it becomes even more sensitive, of course. And there, the dynamics are a bit different. But if we think about single cells first, really the trick is to try to freeze them in a way that doesn't encourage ice nucleation. And typically the way that's been done is to use liquid nitrogen or something very quick, or to some cells, it doesn't matter too much. Like I said, some cells it really does. There are issues with all techniques in freezing. Some of them are safety related. Some of them are sustainability related. Anyone who's used liquid nitrogen will know that it's not a good idea to dunk your hand in there. If you try to work in an undergraduate teaching lab, then if someone can do a dumb thing, they will. So eliminating that possibility for harm is useful. Therefore, there are other electrical ways of freezing things. But even more tragically, people have been asphyxiated in labs by evaporation of liquid nitrogen. So nitrogen is a gas, you can't smell it, you can't see it, but clearly if the air is full of nitrogen, it's not full of oxygen, and therefore you can have some really serious problems. So therefore, the development of other freezing techniques has come along, and there's a whole bunch of those. You can use dry ice and ethanol. You know, they don't have to be sophisticated. You can put things into the -80 lab freezer with something which delays the freezing so that it takes place more gradually and have a controlled rate freezer that way. The most sophisticated way of doing it really is to have electrically controlled freezer, which is gradually reducing at a rate that you can control, pause, reverse the temperature of that sample from typically either 37 degrees or room temperature, somewhere in those ranges, down to the levels below which you can put it into long-term storage. And that could be -80 freezer, it could be liquid nitrogen, it could be whatever. But that process is typically one where it's been kind of overlooked in biology. The sexy stuff is grow the cells, do the manipulations, do the DNA stuff, do the biochemistry and all of that kind of thing, and then put it in a microbial and then freeze it. That's a one-word process. Actually, if you take a bit more care, then it repays. And the same is true going the other way. So thawing is also an important part of the process, it turns out. So however carefully you freeze, if you're rough in thawing, then you can also cause problems, not only by the biophysical problems of the ice, but also you can contaminate quite readily. If your method of thawing is putting in a water bath and the previous person was careless in their hygiene, then you can introduce mycoplasmas or any other contaminants into your cell line just by splashing them around in a water bath that's been not well cleaned. So there's a whole bunch of variables at play, but I think it comes down to people realizing that temperature is as important as any $1,000 reagent that they put into the assay. You need to know that you're doing the same thing the same way every time.
Yeah, if you think about the freezing and thawing process, it's quite a thing actually we're doing. We're going from room temperature or from 2 to 8 down to very low temperatures. And if you look at the physics, there are a lot of things happening while you're doing that. So now, if you look at these actually highly complex process, what are the main parameters we should look after to make sure that once we thaw the cells, they're as healthy as before?
Yeah, and there's a number of transition points in that temperature profile, and they tend to, some of them are biophysical, some of them are sample specific, and it's really down to just a little bit of trial and error sometimes as to figuring out what the best freeze-thaw profile is. And that's where an electrical freezer and thawing device is helpful because you can change that exquisitely. You can change the rate, you can change the stop and start points. So in other words, you can take it down to -10, have a pause, take it down further. All of those kind of things become possible in a way that they're not if you use a biophysical alcohol-based or liquid nitrogen-based freezing. So that's one point of experimenting to find the right points in your cycle. But typically, there are various points in the sub-zero area where things become vitrified or glass-like. And depending on your particular sample, then that can be really important because you want to get through those points safely so that the thing doesn't explode. So you can go from individual water molecules to a kind of glass-like substance without having great big ice crystals in there. So that transition point is really important to know how that works. Once you're past that point, typically you're out the danger zone and you can then get colder and then put it into the storage. Particularly if you're using things like organoids or assemblies of cells, whether it be embryos or anything else, then that vitrification is important to really make sure that the structure of that multi-cell mass is maintained. You can't put an embryo in and get a blob out. That doesn't work. You have to be able to get the same thing out that went in. So it's a very involved and detailed field. And there's lots of experts. There's a group called ATP Bio in the US who really, that cryopreservation is their thing. That's what they do. It's a multi-research group conglomerate who look at this all day, every day. And they've come up with some great technology.
Yeah, speaking of technology, Mark, what are the technological advancements that enabled us to make this process better, more efficient, or technologies that yield better cells at the end once we thaw the cells?
For me, it's one of control. We've known how to freeze things down to very cold temperatures for hundreds of years now. It really comes back to the point I made about treating the temperature profile as like any other aspect of your assay. If you have variability in the way you do that, then you will get variability in the results. And the way that modern technology can help is that, for example, a controlled rate freezer, of which there are several companies who make those with different technologies, but the fundamental point is that it's electrical. So the rate of freezing is electrically controlled so that you don't have to rely on biophysics and guessing and hoping that the temperature profile is what you need. You can actually control it and stop it. So that means that you can have the same machine doing different things to different cell lines or different sample types. So it's quite a versatile way of doing it. And the whole thing just plugs in to the wall. It's not using any chemicals. It's not using any gases. The one that we make uses a thing called a Stirling engine, which is really just a way of electrically compressing and releasing gases in a way that generates a temperature gradient. And therefore you can cool a plate, whether it's a plate full of cryotubes or it's a flat plate with a 96 well assay on top, with incredible control and really make a big difference. And thaw as well. And like I said, thawing is equally important when you take the sample out. then you need to be able to do something with it in a way that's the same every time.
So what I'm hearing, Mark, it's the most important thing is that you do it in a controlled fashion, and the technology itself is less important. So you can achieve that by using different technologies, right?
Yeah. Clearly, if this was a different nature podcast, I'd be selling the hell out of our own system. But, there are other games in town. The way that I think to approach this is to think about, what do I want to achieve? If you have a cell line that's made of house bricks and it doesn't matter what you do, it's going to survive, then yeah, you can be rough with it and cheap in your solution. If you have a gene therapy cell line that's quite fragile and each vial of product at the end is going to be $10,000 and 6 weeks worth of work, why on earth would you throw it in an ethanol bath or something that is not easily reproducible or documentable even? That doesn't really make sense. So it's picking the right technology for the outcome that you need. And typically for research-based experiments where you need to know the subtleties, or for biotech production where you need to be able to do it at the same time every time, then it's worth investing in those extra technologies. If you're just working with spores or something where they're literally going to be there whatever you do, then there are other ways of doing it. But I think that whole emphasis on dark labs is going to have an impact on this as well. We're in an era where the days of an army of PhDs and postdocs doing manual work and just doing the same experiment with a variable change day after day after day, those are probably receding. And now we see a lot more emphasis on, how do we automate that? How do we get that so a machine can do it, so that I can do the interesting stuff and I don't have to stand there with a pipette filling tubes and freezing tubes and doing that kind of thing. So the more control you have in the system, and also the fewer liquids is also important, the easier it is for machines to put things from A to B, and electronics can control the process. You don't want to have liquid nitrogen sloshing around a robot. You don't want to have ethanol around electronics. So that there's ways and means that electrical solutions help you to do then the next task, which is to increase throughput, reduce variability, and so on.
Yeah, that's a great point you're making, Mark. And let's zoom out a bit, because once you have the technology and once you manage to do this in a controlled fashion, it's also about integrating that into a more global workflow. So let's look at the lab workflow. And finally, what it comes down to, we want to prevent errors, we want to prevent variability, and obviously we want to guarantee the quality at the end of the day. So what are your thoughts and suggestions around the lab or around the management itself?
Yeah, and I think that's the big area. Like I said, every time I go to a cryopreservation meeting or even just a general high throughput assay meeting, there's a new advance in robotics. There's new ways to link these things together. I think we're still not there. There's still interoperability issues between instrument manufacturers and instrument types, but we're definitely way, way ahead of where we used to be. I think that the way to think about this is, again, moving backwards from the workflows. What do you want to achieve and what budget do you have to achieve it? Clearly, if you are a small research lab and you don't have much money, then that constrains your choices. If you're a large pharma company and you need reproducibility and quality, then you can afford to pay for that. But the trends in that automation are going to continue because it releases people to do the more interesting thing. And therefore, the technology trend is always going to be, how do we bring reproducibility and automation to that process? So therefore, the 96 well plate is a good example. That's kind of the industry standard for small volume assays now. If you have a robot taking the plate from A to B, it's less likely to trip over and drop it. I was in a lab the other day when this very thing happened. A technician was carrying a stack of 7 or 8 96 well plates, and she tripped and fell over, and one of them tipped on the floor. Now, luckily, they were adherent cells, so all was not completely lost because the cells were still stuck to the wells. But you get my point that humans screw up, and that's just the nature of being human. So bringing a robot into that situation where you already know what it is you need to achieve and the parameters that are going to help you do that, you just want to make it the same every time. I think that's where having a modular approach to equipment in the lab, where everything can play with that robot, is the thing of the future. So that's why, for example, our device is top loading. So a robot hand takes off the lid, puts on the head, screws on, takes off, etc. So it's kind of ready, manual, but ready for robots.
So what you're saying, Mark, is actually an end-to-end automated solution is the go-to solution, right? To make everything automated from A to Z.
I think that's going to be tricky for research labs anytime soon, because clearly that comes with a price tag. But I think anybody who has a process that they do more than a couple of times should be thinking, how do I take variability out of that? And that's the nature of science, is to try to figure out the important variables and isolate everything else and keep it rigid. So if you can find a way to say, it's worth me investing in this technology because it's going to mean that I have tighter data bars, if you like, then that's going to be worth doing. And I think the price of doing that will come down over time. No longer do robots take up whole gymnasiums. Now you can get very effective modular small robots that will help to do tasks that you need in the lab. And like I said, freeing up you, the researcher, to do something a bit more interesting.
And how would that play out for cell therapies? Because oftentimes the volumes are much smaller and it's very individualized. Are the technologies still the same? It's just different cell types, or would everything change, the whole workflow?
I think workflows are always going to be different dependent on volume, but these days, the accuracy of pipetting, the accuracy of the plastics that you use in the machines is very high. The machine learning and the machine vision now that's built into automation devices is amazing. So back in the day, when I saw the first automation equipment, it was programmed based on a theoretical map of the world. So in other words, there was an XYZ position and hole number 7 was in a known coordinate place. Therefore, everything had to be rigidly held in. You couldn't move anything. And often the tip would just hit the plastic top because it was missing slightly. That's not how it's generally done now. The machine vision has a look at where the stuff is, makes a mental map, and then it sends the arm to exactly that point because it's already double checked that it's where it thinks it is. That flexibility to work with different formats of product is going to be huge, because then it means that you can put down something that the machine hasn't seen before and it will adjust itself to the new reality rather than spending days reprogramming all of these coordinates that you would have had to do in the past. So that flexibility of workflow is going to be a real move forward in laboratory equipment generally. There's still going to be a need for the bread and butter type of thing. We make a lot of products that probably haven't changed in 20 or 30 years. Tube rollers, vortexes, stirrers, those are still needed. But I think once a process gets a little bit more high-tech and automated, then you have amazing flexibility nowadays that we just didn't have when I was in the lab.
Hey, smart biotech scientists. I hope you've been enjoying our conversation with Mark Davison, who is the CEO of Grand Instruments. We are continuing our conversation about cryopreservation in part 2 that will drop later this week. I hope to see you there. And tell us, what do you think of this episode? You can do so by leaving a review. And whenever you are ready, I can help you master bioprocesses in 3 different ways. Can help you get clarity. Usually we do this in a few weeks. We can help you craft a winning roadmap so you have the confidence that this roadmap will deliver. We usually do this in about 3 months. And finally, we can also help you collaborate with other industry players so you can 10x your business. If one of the 3 resonates with you, just book a free call using the show notes below, and I look forward to seeing you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.